Welcome back. Today we're going to be looking at how to take proteins and analyze their transmembrane regions and cellular location using PSORTb and the SOSUI server. So the example I've got for you today is actually a hypothetical protein from Staphylococcus aureus. So I've gone ahead into the NCBI protein database, looked up my protein of interest, and here it is. I don't need any information from it other than the FASTA sequence. And so once I have my FASTA sequence, I can go ahead and copy and paste it over to the SOSUI server. Now notice it's got a very complicated URL, so make sure you search for SOSUI, and it's this www server from a Japanese lab. Okay. And then for PSORTb, I sort PSORTb protein, and then I come up with this PSORT.org, PSORTb, that's what I'm looking for. Okay, so first we'll start with PSORTb since this is where I am. You're going to choose your organism type. In this case, I'm dealing with a bacteria. If you happen to know what its gram stain type is, put that in here. You would use advanced, for example, if you're doing tuberculosis. Other than that, positive and negative gram stains should remember from microbiology. You can send the results via email. You can have them in various formats. I usually put this on the web. And then you're going to copy and paste the FASTA sequence in. Oops, helps if I do that. There we go. Okay. And we can upload files if we choose to. You can scroll on down and click submit. No, shoot, I forgot to include the header. I need the header too. So take my header, copy it on over. Okay, still a bacteria, that's positive. Nope, that's if I'm advanced. I'm not advanced. Well, you know, this organism is advanced anyway. There we go, click submit. Well, these usually run pretty fast, which is helpful, but I like doing multiples at a time. So when I come over here, I can take a look at what's available. The SOSUI system is what I'm looking for right there. I can give my job a title if I decide, or I can go ahead and put my sequence in, but notice for this particular case, I do not include the header. So you do not include the header if I'm doing SOSUI, but I do include the header if I'm doing PSORTb. I'll go ahead and execute my query. And notice how quickly the results came in. So you should see total length and your average hydrophobicity score. This is similar to Gravy from XPASI ProPramp program. That's a tongue twister. If these are not similar to what you get from XPASI ProPramp, you have a problem and you need to figure out what that problem is. Notice that this particular example that I have has 12 transmembrane helices. As you scroll down, you'll see where your start of the transmembrane helices is, any amino acids involved in that transmembrane region, where it stops, and then what its length is. You can see variations between lengths for transmembrane regions, but it's helpful to know where they're located within the protein itself and what amino acids are involved. You can scroll down. Sometimes the SOSUI server will give you more hydropathy profile information, but a lot of this I prefer XPASI for, so just something to keep in mind. So this protein does have quite a few transmembrane regions. If I go and take a look at the PSORTb data, now all of a sudden I have various places. Okay, cytoplasmic membrane. These programs couldn't figure out where my protein is located. Cytoplasmic membrane, you get the idea. What's important are the localization scores. So a localization score of 10 is the highest that you can get, and then a localization score of 2.5 is the lowest you can get. Notice how the localization scores are divided between cytoplasm, cytoplasmic membrane, cell wall, or extracellular. If I do not know, or I should say if the program is unable to predict where your protein is located, you will see 2.5 for each of these areas, and then the final prediction will end up being unknown. Fortunately, this protein is rather straightforward because it has these transmembrane regions that SOSUI was able to detect. PSORTb was then able to pick up on those transmembrane regions also, and then proceed to give basically 100% confidence that this particular protein example comes from the cytoplasmic membrane. Now, we expect it to come from the cytoplasmic membrane because SOSUI found these transmembrane helices. Frequently, when there are no transmembrane helices identified by SOSUI, PSORTb will localize, or I should say, claim the location of the protein as either cytoplasmic, extracellular, or cell wall. Simply because to have a membrane-based protein, you need those helices in order to anchor the protein into the membrane. So when you're doing your hypothetical protein projects for class or what have you, make sure that you take a look at the PSORTb results and the SOSUI results together. Because if you're finding transmembrane regions with SOSUI, but PSORTb is not picking up on it as a cytoplasmic membrane protein, people are going to have questions, and you're going to have to be able to dig in a little more to answer those questions. Frequently, though, as I said, they correlate with each other, and so then you can nicely discuss those results as a unit. Please let me know if you've enjoyed this video or if you have any comments or questions. You can do that by emailing me at laura.harris@davenport.edu, or you can go ahead and put comments below. Thanks, and I hope to see you next time. Have a great day.